Last week, there was a question about the inheritance patterns of Rhesus factor. As you might remember from the past, we are familiar with the fact that A and B antigens are on the surface of a red blood cell, so erythrocyte. With the Rhesus factor, the idea is exactly the same. You can consider Rhesus factor as an additional factor that is either present or absent on the surface of the red blood cells. So here we have our red blood cell and a diagram representation of the Rhesus factor antigen. It is a protein that may be in fact a membrane transport protein, but there is a fair bit of current debates about what the Rhesus factor actually does in terms of biochemistry. You might remember that we had a question about that, and for this reason I kept my answers vague. For the purposes of ours, let's just consider that there is on the surface of the red blood cell, just like we saw with the A and B antigens, or sprinkles, as you might remember from my donut analog. So it is either present or absent. You might remember that there are many subgroupings to the Rhesus factor. The last time I checked, I believe it was 52 or something like that. But the one that we are going to be interested in for our purposes here as an antigen is the D factor. So for the purposes of our class, when we're talking about Rhesus factor, we're really talking about the D factor. So it is important for us to think about how this factor comes about to existence on the surface of a red blood cell, and it is entirely genetic. So do you remember the Mendelian inheritance patterns? Rhesus factor, so our D antigen, is coded by a dominant gene, and it is located within the chromosome number 1. Here, I have made a diagrammatic representation of that. We have homologous, so the same pair of chromosomes, and the Rhesus factor is going to occupy one of the positions on that chromosome. So this gene in this position contains the genetic information required to synthesize the Rhesus factor. Now, remember that a dominant gene is the one that will be expressed if present. And Rhesus factor is coded by a dominant gene. So by convention, we will denote the dominant gene by a capital letter. And the recessive gene, which would result in no Rhesus factor present, we will denote that by a lowercase letter. Now, I kept it very simple in our last lecture, so this is going to be an expansion from that. Okay, if there is a Rhesus factor incompatibility problem leading to hemolytic disease of the newborn, that always occurs with the Rhesus negative mother. Remember, in the US, about 15% of the population are Rhesus negative, so it would be reasonable for us to expect that 15% of the women that we get giving birth are also Rhesus negative. Interestingly, in Asian populations such as Chinese, the probability is as low as 1 to 5% for the women that are going to be Rhesus negative. But here, for us, it's about 15%. Now, here we have a homologous pair of chromosomes with the relevant gene. So if the mother is Rhesus negative, that has to be that she does not have the Rhesus positive gene. So we will know for sure that if the mother, or anyone for that matter, is rhesus negative, they will have two lowercase r's there. They will be both recessive genes that we see here. But if someone is rhesus positive, then one or both of these genes are going to be dominant. So here we see someone with two dominant copies of this gene, so they're going to be rhesus positive. And if someone has heterozygous copies of the gene, so capital R and lowercase r, the gene for the rhesus factor is going to be expressed. So this will allow us to do some calculations or genetic plans, if you wish, to predict the probability of the rhesus factor incompatibility occurring and resulting in the hemolytic disease of the newborn. This is only going to happen with the rhesus negative mother and rhesus positive father. Let's map it out. So we know that the mother is going to be rhesus negative. So we have two lowercase r's. This means the mother is homozygous recessive. And we know that the father could be one of the two options. One capital and one lowercase r. So he would be carrying one copy of the rhesus gene, while the other one is recessive. In other words, this father is heterozygous for this trait. Now, to reproduce, the mother will make an ova, an egg cell, and the father will make sperm. And now we can map out all the possibilities of how these can recombine. So one possibility would be these combining, and that would give us a rhesus positive fetus. Notice a lowercase r and uppercase r. Alternatively, these could combine, and that would give us a rhesus negative fetus, lowercase r and lowercase r. Or it could be these two. And again, this would result a rhesus positive fetus because there is one copy of the rhesus gene present, which is of course dominant. So lowercase r and uppercase r. Or it could be these two combining. This would give us two lowercase r's, so a rhesus negative. So here we see that the probability is 50-50 that any given fetus is rhesus positive. The presence or absence of the rhesus factor is entirely genetic. Alternatively, we know that the mother is going to be rhesus negative, but the father could be alternatively homozygous for that trait with two rhesus positive genes, so two capital R's. Phenotypically, the father is still, of course, rhesus positive. In this case, the mother produces ova and the father produces sperm as such. Let's map out the combinations here. So these two could combine, giving us lowercase r and uppercase r. So we have a rhesus positive fetus because there is one rhesus positive gene. Actually, it is the exact same possible combination that we see here. And again, likewise with this one, and indeed the same even here. So in this case, even though the mother is rhesus negative, 100% of the potential fetuses formed by this union would be rhesus positive. Therefore, probability of the rhesus factor incompatibility occurring is a relatively high one. So rhesus factor is really quite simple. It's either there or it's not. It's coded by a single dominant gene which may be present in homozygous or heterozygous form in the father. So consider rhesus factor incompatibility whenever we have a rhesus negative mother and a rhesus positive father. And it's for me this time. I appreciate you joining me.